Less than 100 years ago, if you got an infection, your chance of dying is what we would consider in today's terms unacceptably high. This is a future we might be facing again. Grouping all deaths together, infection is a leading cause of death. However, over the past few decades, we've seen a fantastic trend in decreasing mortality due to the leading infections. TB, as an example, has been on the decline year on year. This is in no small part due to global initiatives to improve treatment options, reduce the spread of HIV, and improve sanitation and nutrition. However, there are still monsters lurking over the horizon. Our treatments are becoming less and less effective. One of the most cited opinions on the future of antimicrobial resistance is the O'Neill report. Unlike a lot of other research on the topic, O'Neill was tasked with predicting what the future might hold for us, what steps we might take to mitigate the issues. The largest takeaway is that by 2050, I am more likely to die of an infection than I am of the current second most common form of death, cancer. If you're watching this now, the same applies to you. The cause of your death is more likely to be a once treatable infection rather than cancer. Having lost several loved ones and friends to cancer, a fate I know I'm not alone in, this is just mind-blowing. Several academics, myself included, have technical issues with the O'Neill report, but nothing pushes me really to say its outcomes are flawed. But 2050 is a long way off, right? And the numbers might be a little exaggerated or they might be wrong. Well, that's great for future, James, but AMR is a problem now. There are people who will die today from a disease we could have treated in the past. To put some numbers in context, COVID at its current height killed 3.5 million people in 2021. I'm sure you don't need me to tell you this was a global problem. How does it compare to our rising issue? In 2014, the number of deaths due to AMR was estimated to be around 700,000, but in just 5 years, that number almost doubled. 1.27 million people died due to an AMR, antimicrobial resistant, infection. Another 4.9 million died with an associated AMR infection. The bacteria may not have killed them, but it certainly didn't help for those nearly 5 million people. So how did we get to this point? Humanity's been trying to combat infections for centuries. One of my favorite examples was Bald's eye salve from the Leech book. This was a Middle Ages concoction to treat eye infections. Researchers in 2014 showed that by making the salve as described in the recipe, with some translation from Old English or Middle English into current English, you could kill common eye pathogens like Staph aureus. You might also recognize a familiar name in the acknowledgements of this paper. Other more extreme methods of treating diseases were tried throughout the years. Mercury rose to fame as a potential treatment for a new plague sweeping through 16th century Europe called the French disease. To some people, we now know it as syphilis. This led to the term a night in the arms of Venus, a lifetime on Mercury. Physicians correctly surmised that the disease was contagious and spread by some particle during intercourse. In non-lethal doses, mercury induces salivation and increased urination, and it was thought by the physicians of the day that this would expel the disease particle in the saliva and urine. Doctors recommended enough mercury to produce roughly 3 liters of saliva. There can be no doubt, at those levels the dose of mercury was toxic. Silver nitrate was another pre-antibiotic with dubious effect and higher risk. In order to treat gonorrhea, silver nitrate would be pumped up the offending member until morale improved. Both chemicals likely did have some minimal effect on killing bacteria. They're both really toxic. It wasn't until the start of the 1900s that we started thinking about more effective antibacterials. Salvicin was one of those drugs. It was discovered in the lab of Paul Ehrlich. His great idea was to screen hundreds of chemicals for those that could kill a pathogen like a bacteria or a fungus, but have little effect on us. He termed this idea magic bullets. Salvicin was far less dangerous than mercury compounds, which were still in fashion at the time. It was actually an arsenic compound itself, and it was highly effective against syphilis. But it wasn't the most stable chemical, requiring meticulous handling and preparation, which really limited its ability to be used widely. A paper published in 1929 by Fleming, which was somewhat ignored until an Australian and German chemistry team of Floley and Chain worked out how to produce a bunch of it. This would become the first true antibiotic, penicillin. Penicillin is a natural product made by fungi, probably to help them fight off bacteria invading their space. In its early years, it was fraught with failures and issues around production. During the first trials, there was so little penicillin that researchers had to extract it from the urine of the patients who were receiving it. But how does it actually work? We know today that penicillin interferes with how bacteria put together their cell walls. Bacteria cell walls are made up of several different structures. There's an inner membrane, a peptidoglycan layer, and if you're gram-negative, an outer membrane. But we're interested in the peptidoglycan layer here. Penicillin binds a protein that helps build this structure. Scientists, because they're great at naming stuff, called this protein penicillin binding protein. Penicillin sits in the enzyme, stopping it from being able to add more to the cell wall, which leads to the cell dying. But resistance to penicillin was not far behind. In fact, Fleming even mentioned it in his Nobel Prize acceptance speech in 1946. But for the first time, we had a relatively safe molecule you could just swallow as a treatment, and it would cure your infection. But it wasn't to last. Bacteria became resistant. In fact, they probably were already resistant even before we began treatments. One way they are resistant, or can become resistant, is by the production of beta-lactamases. This is an enzyme, hence the name A's at the end, that breaks down beta-lactams, which are drugs like penicillin. Penicillin's success, however, and our better understanding of biochemistry and chemistry led to a boom in the development of novel drugs. But this timeline has a shadow. Each and every drug discovered was soon followed by the emergence of resistance. No problem, as long as the pipeline doesn't dry up. Between 1970 and around 2000, no new classes of antibiotics were discovered. Plenty of derivatives with really cool modes of action were developed and made and helped a bunch of people, but no new classes of antibiotics were discovered. Outside of my phage work, another topic I work on research, which I'll hopefully publish a paper on soon, is adjuvant therapy. If you've had any dental work in the past decade or so, you've probably been given a drug called coamoxiclav. This is two drugs, actually. It's an antibiotic and an adjuvant. The antibiotic works as we expect, like a normal penicillin-type antibiotic. However, the adjuvant looks a lot like the antibiotic, but doesn't kill the bacteria. Instead, what it does is it binds the beta-lactamases, stopping them from being able to degrade the beta-lactam, the thing that will kill the bacteria. But to get around this anti-resistance drug, bacteria develop resistance. They can titrate out the adjuvant by increasing the expression of beta-lactamases, or they can literally just pump it out of the cell. This is the state of current drug discovery and clinical use. For each new drug we develop or steal from bacteria or fungi, they find a way to fight back. We are in an arms race with bacteria, and they are much better chemists than us. This brings us back to the ranch. Today, antimicrobial resistance is a real problem. Millions will die over the next decade from infections my parents' generation could treat. Millennials really are the most cursed generation. So what can we do? Back to the O'Neill report. It was different from other papers, one, because it was commissioned by the UK government, but also it was written by an economist. They suggested that the problem isn't a lack of potential drugs. Out there in drug space, there are an infinite number of drugs and one of them will work. Instead, it's about economic incentives. Say we're a big pharmaceutical company. Outside the billions we spend on advertising, we occasionally spend some money on developing new therapies. To bring an antibiotic to market would currently cost in the order of about $1.5 billion. Imagine we develop a new drug. There's no inherent resistance, it's safe, broad spectrum, so it works on loads of infections. Yeah, we spent $1.5 billion on it, but in the grand scheme of things, that's not a huge chunk of money if we're talking about saving millions of lives. But the issue is this: what would happen to that drug? It would be left on a shelf as a last resort. Rightly so, I guess. No doubt, if it was used a bunch, we'd see resistance become rampant and we'd be back to square one, holding our pipettes in our hands. So how do we recoup our investment? This is a problem for politicians, lawyers, and economists, something I am not qualified to do. But the O'Neill report had four main takeaways: public awareness, improve the supply chain, reduce the use in humans and animals, and somewhat related, reduce the use in agriculture. Those four things can be done. I also like the suggestion that we should pay microbiologists more. Thanks, O'Neill. Those are all things we can do, but not individually. Much like our friends, the climate scientists, we are in a position where our individual actions have little impact on the issue. If your doctor recommends you take an antibiotic, you should 100% take it. But we should be putting pressure on governments to step in and address the real problems. No amount of lab-based research can solve this problem. We already have the ammunition to fight this problem, the magic bullets. But without coordination, we will lose this fight. Thanks for watching. Well, I hope you enjoyed that. If you don't know, if you didn't see the video earlier, I made a catch-up about where I've been out. So it's been 7 years. Basically, I finished my postdoc in France, moved to the US, did another postdoc here, became an assistant professor, now have my own research group doing phage therapy. This video was largely inspired by Simon Clark, Dr. Simon Clark's Nebula series about science communication videos. It enthused me to go and make another one. So you have him to thank for it. And if you want to thank him, why not go check out his channel, link down below. It's really great climate science stuff and a whole bunch of other really cool stuff. So that just lets me say thank you for watching. I'll see you next time. As I said in the catch-up video, I've got a couple of these in the bag, so you'll see them soon, hopefully. Thank you. Bye.